Thank you very much for this kind introduction. Thanks also to the scientific and the organizing committee for having invited me to participate in this conference. It's really, really a pleasure to be here today. It's my first time at the EUCARPIA meeting, and I hope that it won't be the last one. And I would also like to congratulate the organizing committee for this conference. You have done an amazing work. So from my side, thank you very much for all this work. Well, the talk I am going to present, or the work I'm going to present here today, it's about modeling and estimating genetic effects in high-throughput phenotyping data using a spatio-temporal model. This spatio-temporal model, will allow us to describe how genetic effects evolve over time, while at the same time accounting for the spatial variation which is commonly present also in this kind of experiments. Of course, this is not, This is not a work that I have done alone. Many people have contributed, and here you have some of them. I believe that some names are really familiar to you. So we have Fred van Ewijk and Martin Boer from Wageningen University and Research. Both of them are here today. Then Andreas Huhn from ETH Zurich University in Switzerland, and Lukas Cronenberg, who is now at the Humboldt University in Berlin. And finally, we have Diana Perez. Diana Perez is a statistician from Colombia, now based in Spain. And Diana did her PhD under the supervision of Fred van Ewijk and myself on the analysis of high-throughput phenotyping data. So the work I am going to present here today is actually part of her PhD dissertation, so the credit should also go to her. So this is an overview of the talk. I will start with a very, very brief background about high-throughput phenotyping. Then I will describe the type of data that we will be dealing with. As illustration, I will use data from an experiment conducted at the field phenotyping platform located at ETH Zurich University, and I will also present here the model that we consider for the analysis of this data. As you may expect, p-splines play a fundamental role in our modeling strategy, so in the third part of the talk, I will briefly describe p-splines as well as their connection with linear mixed model, which is actually what we use for estimation. Then if I have time, probably not, I will come back to the model to present some extra technical details, and then I will finish with some results for the FIPS platform. So let's start. Why do we need, why do we need high-throughput phenotyping? Well, in the past decades, plant genetics has advanced, I would say, at extraordinary pace. Sequencing is now fast, it is affordable, and almost routine, which means that for many crops, we can now generate vast amounts of genetic information with relative ease. However, by contrast, plant phenotyping that is measuring observable plant traits has advanced much more slowly. Traditional methods are commonly manual, slow, time-consuming, and labor-intensive, and perhaps most importantly, they are in many cases destructive, which makes it difficult, if not impossible, to measure plants repeatedly as they grow. So this mismatch between genotyping and phenotyping, it's often referred as the phenotyping bottleneck, and it actually limits our ability to link genotypes, phenotypes, and environment. So as a response to this bottleneck, to this challenge, in recent years, we have seen the rise of high-throughput phenotyping techniques and platforms which use a wide range of sensing equipment to allow or to enable for the almost continuous monitoring of plant phenotypic traits. And there is now a broad spectrum of high-throughput phenotyping platforms ranging from controlled environment system in greenhouses or growth chambers, such as, for instance, the PhenoArc platform in INRAE, Montpellier, to field-based platforms equipped with drones or cable-suspended system, which is, for instance, the case of the field phenotyping platform at ETH Zurich. Well, this image from the paper by Moreira and colleagues somehow illustrates the idea. So here we see a plant, and we see how this plant evolves over time, how this plant grows. And then we have a drone, which by means of different technologies is used to measure different characteristics of this plant all along time. So as a result, we now have access to possibly, although not necessarily, long time series of repeated measurements of plant phenotypic traits between, let's say, the emergence of the seed and the physiological maturity of the plant. So the key point here is that high-throughput phenotyping the platforms provide which is non-destructive and often more cost-effective than traditional manual methods. Of course, high-throughput phenotyping platforms can generate many different types of data. This is one of their strengths, but in this talk I will focus on a particular type. So before going further, I would like to introduce the type of data that we are going to deal with, as well as the notation. So as YI, I denote the phenotypic trait at the ith experimental unit. For simplicity, I will refer it as a plot. And this phenotypic trait is measured at times T1, T2, until T capital N. And we have information on capital M plots, all of them measured at the same time. That is, we have balanced data. Then we can have, well, we have capital L genotypes. It's replicated a number of times, NG times, and I will denote as G of I the genotype that is planted at the ith plot. And finally, we also allow for a population structure. That is, the genotypes can be grouped or can be clustered in panels or families of genotypes, and we can have capital K populations, each with a different, possibly different number of genotypes. And as P of I denote the population of the genotype that is planted at the ith plot. Well, all in all, what is really important here, apart from the notation, is that we have a nested hierarchical structure. This is the kind of data that we are going to deal with. So we have plants nested in genotypes, and also genotypes nested in populations. Well, we will also assume that these plots can be mapped to a coordinate system defined in terms of capital R rows and capital C columns. This is needed for the spatial analysis in our case, and I will denote as R of I and C of I the row and column position of the ith plot. Well, to make things a little bit more concrete, I will use data from an experiment conducted at the field phenotyping platform. The FIP platform is a field-based, cable-suspended, multi-sensor platform located in an area of around 1 hectare, divided in 6 different lots. So here you have a plot of the field, the platforms, with the 6 different lots. Here you have 2 pictures of the sensing devices that are part of this platform. So for the experiment conducted in 2017, this is the data that I am going to use. The experiment was conducted in lots 2 and 6. So for our analysis, we needed to create a virtual grid composed of 42 rows and 36 columns, although as you can see here, only 720 plots were actually used. We needed to create this virtual grid for the spatial analysis. Well, a total of 334 genotypes were tested from a diverse panel of European wheat. All of them were replicated. Most of them had 2 replicates, and then there are 3 checks which were replicated between 18 and 22 times. In this case, these genotypes can be also grouped in population, actually in regions of origin, corresponding to the country at which, or where they were first inscribed in the European catalog. And in total, we have 7 different regions of genotypes, each having a different number of, we have 7 populations, regions of origin, each having a different number of genotypes. Here you see a map of the 7 populations. Recall that the experiment is conducted in Switzerland, but as I mentioned, we have genotypes from 7 different regions. Okay, finally, the phenotypic trait was the canopy height, and this canopy height was measured a total of 23 times between the end of February and the end of June. And this is how the data looks like. So this video shows the spatial distribution of this canopy height at the 23 time points at which it was measured. And as you can see, as time evolves, things change. So this is the same information, but now for 4 different times. And as you can see, particularly for some time points, there is a rather clear spatial pattern, but we also observe that this spatial pattern changes over time. Okay, this is a different way to look at the same data. What we see here is the evolution over time of this canopy height for the 23 times at which it was measured for the replicates of 2 different genotypes. Okay, and as you can see, these 2 genotypes performed, let's say, in a different way. With this genotype, these replicates, they grew more. So, as I mentioned at the beginning of this talk, our aim in this work is to model and estimate genetic effects, but in this case, we are interested in modeling and estimating the evolution over time of these genetic effects. However, we need to correct for environmental factors, non-genetic effects, spatial variation, and as we just saw before, this spatial variation can change also with time. This implies that for the analysis of high-throughput phenotyping data, at least the kind of data that we are dealing with, we need a spatial-temporal perspective. And, well, the computational aspect here is something that we should be aware, because we may have not only rather large sample sizes, but also highly parameterized model. In fact, being able to fit the model that we wanted to consider for the analysis of this data was perhaps one of the major challenges of this work. So, which is this model? So we use the SPATS model as the base model. So for a particular time point, the SPATS model for the FIP data will be as follows. So here, FP is the fixed effect coefficient for the population P, region P, with P varying between 1 and 7. We have 7 regions. FG is the random effect coefficient for genotype G. FR and FC are random effect coefficients for row R and column C. And then we have this FS, this 2-dimensional smooth function defined in terms of the row and column position of each plot in the field. So these 2-dimensional smooth functions accounts for the spatial variation. We use P-splines to model it. So if we fit this model for the data collected at a particular time point for the FIP data, this is some of the results that we will obtain using SPATS. As you can see here, it's the spatial distribution of the canopy height. This is the estimated 2-dimensional smooth function, this component here. Okay, and as you can see, we have been able to recover, I would say, relatively well the pattern that we observed in the raw data. This is the spatial distribution of the genotypic blobs. Well, there is no clear spatial pattern in these blobs, which is what we would like to see. I mean, they say the spatial variation have been captured by other components in the model. This is the histogram. So, of course, one possible approach for the analysis of the high-throughput phenotyping data will be to fit a SPATS model separately for each time point, then extract the genotypic blobs and reconstruct the evolution over time of this genetic effect. However, well, in this work, we opted for an approach that allows us to model all data, all time points simultaneously, therefore borrowing strength between different time points. So, well, using the SPATS model as the base model, we extended it to the spatial-temporal case by allowing all these effects to vary with time. This is the model. Well, in this case, just a comment, we say we have enough information to decompose this time-dependent error term into 2 components. Okay, a structure component associated with each plot and somehow capture its evolution over time. We can think about it, if you prefer, as a functional residual. And then we have the pure measurement error. One of the reasons for this decomposition is that somehow avoids the need to model correlated noise, which in many cases, I would say it is far from trivial. So this is the final model that we consider for the analysis of high-throughput phenotyping data. So how do we interpret each of these effects here? Well, Fp of t, it's now the time-varying effect coefficient for population P. Fg of t, it's the time-varying random effect coefficient for genotype G. Fi of t, well, this Fg actually measures the deviation of the effect of the genotype with respect to the population to which it belongs. Fi of t, it's the time-varying random effect coefficient for plot I. It measures the deviation from the effect of the genotype that is planted at that particular plot. Then Fr of t and Fc of t are time-varying, again, random effect coefficients for row R and column C. And finally, we have this FST. This FST is a three-dimensional spatio-temporal surface defined now in terms of the row and column position of its plot in the field, but also in terms of time. So this three-dimensional surface, this spatio-temporal surface, allows or accounts for spatial trend variation, but allows these spatial trend variations to vary with time, which, looking at the data, it seems like needed. Well, so the question now is, how do we model and estimate each of these time varying effects? Well, I will say that looking at the data, it seems reasonable to assume that all these effects vary smoothly along time, so we propose to model these effects using a smoothing technique, in particular using penalized splines, P-splines, so one-dimensional P-splines for all these one-dimensional functions here, and then a three-dimensional P-spline for this spatio-temporal trend, this spatio-temporal surface. But before going into the details about P-splines, let's have a better idea about the type of results that we can obtain from this model and what can we do with them. So first, we obtain the estimated population or region trajectories, the estimated population growth curves, which represent the average growth pattern for the genotypes that belong to that particular population. Then we have the genotype-specific deviations, which describe how these genotypes differ with respect to the population to which they belong. Similarly, we obtain also these plot-specific deviations. So the sum of these two components, population trajectories and genotype-specific deviations, provide what we call the genotype-specific trajectories or the genotype-specific growth curves. Similarly, well, the sum of the three will provide what we call these plot-specific trajectories or growth curves. So what can we do with the results? Well, mostly we will be interested in these two curves, genotype-specific deviations, genotype-specific trajectories, because, well, they are related to the genotypic performance. So what can be done with them? Well, if we focus on these genotype-specific trajectories or growth curves, as well as their first and second order derivative, we can use these curves to compute for each genotype different time-independent characteristics that can be useful in practice to characterize or to summarize the performance of the different genotypes. So for instance, we could compute, let's say, the maximum growth rate from the first order derivative, or the onset and the end of growth, as well as the duration of the growth phase from the second order derivative. Okay, this is from the genotype-specific trajectories, but we can also focus on the genotype-specific deviations. And note that in this case, positive deviation actually correspond to better genotypic performances compared to this average population effect, while negative deviations actually correspond to worse genotypic performances compared to this average population effect. So we can summarize and compare the performance of the genotypes by computing, for instance, the area under this curve, either for the whole interval at which the phenotype was measured, or for a particular time interval in which we might be particularly interested. Okay, well, it's true that up to now, I mostly use the results of this model in I would say, a rather descriptive way, like focusing in these estimated curves as well as these time-independent characteristics to, let's say, summarize the performance of genotypes. And although I haven't explored that myself, well, I believe that these results could also be useful or could also support a range of other applications. So for instance, we could use these estimated curves as inputs in functional regression to, for instance, predict endpoint traits such as yield. We could also try to compare directly the curves to identify when and where these genotypes differ. And we could also use these estimated curves, or the summaries, to derive similarity measures between these genotypes and then include these similarity measures in other models in order to predict other traits. So all in all, although so far I have been mostly using this result in a descriptive way, well, the results of the model could also provide a basis for a wide range of downstream applications. Well, P-splines and linear mixed models. So P-splines are at the core of our modeling strategy, so I really would like to spend some time presenting P-splines. I hope it's not too technical or known. Well, very briefly, P-splines have two main ingredients. So the first thing that we have to do is to represent, actually to approximate the functions that we want to estimate as a linear combination of B-spline basis functions. And typically we are going to be very generous with the number of B-spline bases. Let's say our aim somehow is to ensure that the function that we want to estimate lies in the space spanned by this B-spline basis. Of course, this generosity comes at a cost, the cost of overfitting. So in order to avoid this overfitting, the estimation procedure is modified by incorporating a penalty on the regression coefficient. So to make it simpler, I will start with the one-dimensional case. So in this case, the function that we want to estimate depends only on one covariate. Here it's time, and there are no other components in this model. So in this plot here, the dots, I don't know whether you can see them, are the canopy height. The canopy height at a particular plot in the field, but for all time points at which it was measured. And our interest lies in estimating this blue curve, the growth curve. Well, as I said before, the first thing that we have to do is to approximate, to represent this curve as a linear combination of B-spline basis functions. So this is how a B-spline basis functions look like. And each, as you can see, each of these B-spline basis function is multiplied by a different regression coefficient. Well, under this representation, only the vector of regression coefficient theta is unknown. The model is linear on this regression coefficient, so we can estimate them using ordinary least squares really easily. And if we do so, this is what we obtain. We overfit the data, we fit the noise, because we have chosen a very large number of B-spline bases. So in order to avoid this overfitting, what Ehlers and Marx proposed was to modify the least squares criterion by incorporating a penalty on these regression coefficients. In particular, what they proposed was to penalize the differences of order Q on coefficients associated with adjacent B-spline bases. Let's say the idea of this penalty is to not allow coefficients that are close to each other to go up and down. In this way, you also avoid the function estimate to go up and down, and in this way, you avoid the overfitting. Well, this is in a more compact way. Here, capital D, it's the matrix that forms differences of order Q. P, it's the so-called penalty matrix. And lambda is the smoothing parameter. And as you can see here, lambda controls the weight of the penalty in the minimization problem. So small values will give rise to overfitting, okay, ordinary least squares, while very large values will give rise to very smooth function estimates. Here you see the results using a more adequate value for this smoothing parameter. So the smoothing parameter actually controls the trade-off between fidelity to the data and the smoothness of the function estimate. Of course, a critical issue here is setting the right value for this smoothing parameter, selecting the appropriate amount of smoothness that is needed by our data. Okay, but before going into this, let's move to the two-dimensional case. Well, now the data that we have is the observed canopy height at each plot in the field, but for a particular time point. And our interest here lies in modeling and estimating this surface that you have here. These plots are defined in terms of the row and column position. So when it comes to extend the B-spline principles to the two-dimensional case, the first thing that we have to do is to approximate this two-dimensional smooth function in terms of B-splines. And this is accomplished by the tensor product of two marginal B-spline bases, one along the row direction and the other one along the column direction. But now we need to specify a penalty in 2 dimensions. So before doing that, I think it is worth seeing this vector of regression coefficients in a matrix. So in this case, and as you can see, the rows of this matrix correspond to the regression coefficients along the column direction in the field, while the columns correspond to the regression coefficients along the row direction. And now we do exactly the same as we did for the one-dimensional case. We penalize coefficient differences, but we do that separately for the rows and the columns of this matrix of coefficients. So this is the penalty in 2 dimensions. So this term here forms differences of order Q1 along the columns of this matrix of coefficients. So jointly with lambda 1, the smoothing parameter, controls the amount of smoothness along the row direction, while this term here forms differences of order Q along the rows of this matrix of coefficients. So jointly with lambda 2 is responsible for the smoothness along the other direction. Okay, so this is the penalty in 2 dimensions. Note that we are considering 2 different smoothing parameters, that is a different amount of smoothness along the row and column direction. We are considering anisotropy. Of course, this idea can be extended also to the spatio-temporal case. Now the function depends not only on the row and column position, but also depends on time. So we need the tensor product of 3 marginal B-spline bases for the rows, for the columns, and also for time. And now the penalty has 3 terms and also 3 different smoothing parameters. So this one here is responsible for the smoothness along the rows. This one here is responsible for the smoothness along the columns, and this one here is responsible for the smoothness along time. Okay, but okay, what about estimation? And here is where linear mixed model comes. Well, in all cases, in matrix form, our model can be specified in this way. Here, capital B, it's the B-spline design matrix, possibly obtained using the tensor product if we are in the multidimensional case, and the vector of regression coefficient theta are estimated by minimizing the penalized least squares objective function. So this is the penalty, and P lambda here is the global penalty, the penalty acting on these regression coefficients in order to impose smoothness. This penalty can, in all cases, specify as a linear combination of known penalty matrices, this PL, each multiplied by a different smoothing parameter. This is, this expression here, but in a compact way. Well, it's relatively easy to show that for known values of these smoothing parameters, the solution to this expression actually corresponds to the best linear unbiased predictor of theta under the assumption that theta follows a multivariate normal distribution with 0 mean and variance covariance matrix sigma squared, the error variance, times the pseudo-inverse of this penalty matrix. And 2 things to note here. First, the relationship between the penalty matrix and the variance covariance matrix, which one being the inverse of the other. So we can interpret a penalty matrix in the P-spline setting as a precision matrix. And second, we need to work with here a pseudo-inverse. The reason for that is that this penalty matrix is not a full rank, and the rank deficiency arises from the fact that there is a part of the function f that we want to estimate that is not penalized by the penalty. So one possibility to avoid the need to work with a pseudo-inverse, and the one that we follow in our work actually, is to decompose, to reformulate the model, or to decompose the function that we want to estimate into 2 parts, okay? One part which is not penalized by the penalty, and the other one which is penalized by the penalty, which in the mixed model setting implies that here beta is a vector of fixed effects unpenalized, while u is a vector of random effects following, again, a multivariate normal distribution. But now this variance covariance matrix, or its precision, is of full rank. Again, this precision, recall the relationship between penalty matrix in the P-spline setting, precision matrices in the linear mixed model setting, can be again specified as a linear combination of known matrices, this calligraphic GL, each multiplied by the inverse of a variance parameter, which is the ratio between the error variance and the corresponding smoothing parameter. So this link between penalized splines and linear mixed models have, I would say, many, many advantages, but also there are some complications. In my opinion, the major advantage is, as you can see here, the smoothing parameter are ratios between variance parameters. So setting the right value for this smoothing parameter, selecting the appropriate amount of smoothness becomes a problem of estimating variance parameters. So we can use restricted maximum likelihood then it also makes, I would say, much easier to build up more complex models, including both fixed and random effect, as we do in SPATs, but also multiple smooth functions, which is what we actually need for the model that we are considering for high throughput, for the high throughput phenotyping data. If the model that we consider includes only one-dimensional p-splines, one-dimensional functions, on top of other fixed or random effects, then estimation can be carried out with almost any software and algorithm for linear mixed models. The problem arises when we want to incorporate in our model multidimensional p-spline, multidimensional functions. Okay, then in this case, estimation cannot rely on standard, let's say, mixed model software, so we need specialized methods and algorithms. And this is an area in which I worked in the past. In fact, the implementation of the model that we are considering relies on this sub-method here. Well, now that we have all ingredients we need, okay, let's come back. I think that I have time to talk about the model. Recall, this is the model that we are considering. Recall that we are assuming that all these time-varying effects vary smoothly along time. So we propose to model them using p-splines, that is, using p-spline basis functions, possibly the tensor product, and in this case, in combination with a second-order difference penalty. So for the one-dimensional functions, the reformulation, the mixed model form that we consider is this one. So the fixed part contains the intercept and the linear trend along time. And then we have the pure nonlinear component where these coefficients are penalized or are assumed to be random following a particular variance-covariance matrix. However, for the effects associated with genotypes, plots, rows, and columns, we also opted for to penalize these unpenalized coefficients, intercepts and slope. That is, we changed their status from fixed to random. I mean, there are several reasons for that, and one of the major reason is that without these extra penalties on intercepts and slopes, then the design matrix associated with the fixed or the unpenalized part of the model wouldn't be of full rank. Okay? Also, it aligns, in my opinion, better with the longitudinal data, data literature. Well, with respect to the spatio-temporal three-dimensional surface, we use a tensor product of three marginal b-spline bases in combination with an isotropic three-dimensional penalty. It also accepts a mixed model form, which in this case, it's convenient because it allows us to detect redundant components in our model, because we also have this population effect. So when we construct these design matrices, we simply remove the redundant components from this specification. Okay, well, all in all, at the end, what we have is a linear mixed model, a rather complex linear mixed model, but still a linear mixed model, or at least we can see it as a linear mixed model. And here you have some computational aspects that we needed to be able to fit our model in reasonable computing time. So first, we exploit the sparse structure of the design matrices. Our code is pure R, and then we use the span and matrices R packages. Then we also exploit the fact that the data we have presents an array structure. We are considering, as I mentioned, balanced data. So it allows us to use generalized linear array models to construct the cross product involved in Henderson equations. We also use the sparse Cholesky decomposition for solving these Henderson equations, and regarding the estimation of the smoothing parameters, the variance parameters, we use the sub-methods in combination with some functions and methods derived by Martin Burr and implemented in the R package LMM solver, which is actually very computationally efficient. So let's now go to the results for the FIP platform. Well, regarding the model specification, for the one-dimensional smooth functions, we considered b-spline basis of dimension 20, And for the three-dimensional spatio-temporal surface, we considered a marginal B-spline basis of dimension 8. Well, with this specification, the model has more than 23,000 coefficients and 22 smoothing parameters, variance parameters. So the model needed around 10 minutes to be fitted. I mean, it's true that this computing time is using my lab, my PC at the office, which is a rather, I would say, good one. If I use my laptop, this computing time will increase up to around 30 minutes, which is, of course, longer, but I will say that this is still reasonable. As I mentioned before, one of the major challenges here was to be able to fit the model in, I would say, reasonable computing time. Still, there are some computational aspects that need to be considered. Well, so first result, this plot shows the observed canopy height for 4 different plots. These are the dots, the color dots that you see here, and the color lines. The dotted line is the fitted values. And as you can see, we have been able to recover, or the model have been able to recover relatively well the observed data. This is a result similar to the one that we obtained with the SPATs. Here you see the spatial distribution of the canopy height for 4 different time points. Here is the estimated spatio-temporal surface, and as you can see, particularly for some time points, we recover well also the pattern that we observe in the raw data. These are perhaps the most interesting results. So this plot here shows the estimated growth curves for each region, for the 7 regions that we have. This is the estimated first-order derivative, okay, where we have highlighted 3 different local maxima. And as you can see, there are differences between regions, both in the growth curves, in the growth patterns, but also in the growth rates. So for instance, if we focus on the growth curves, we see that particularly at the end, the regions seem to cluster according to the region of origin. So this map here shows the 7 regions with the 3 different clusters. So this clustering may perhaps indicate physiological adaptation to different climatic regions in Europe. Well, this gray line that you see here represents the average temperature across the whole experiment. Okay, and as you can see, there was a very cold period. Here it is April, where, as you can see, growth slowed down, and another one around May. So we could, in principle, interpret the 2nd and the 3rd local maxima in this growth rate as recoveries after this too cold period. So we can link the results that we obtain with our model also with environmental variables. These are the results at the genotype level. So on the top, we have the genotype-specific trajectories. In black, the black line is the region-specific trajectory. As you can see, and as expected, this region trajectory summarizes well the performance of the genotypes that belong to that particular region. And on the bottom, we have the genotype-specific deviations. Again, recall that positive deviations correspond to better genotypic performances, while negative deviations correspond to worse genotypic performances. And as you can see, and as expected, I mean, the magnitude of these deviations, and therefore the difference between genotypic performances, increase in all cases with time. Well, I had another slide here with some time-independent characteristics that we can extract from these curves. I will skip it and then go directly to the conclusions. So in a previous work, we showed the SPATS model that PSPAN may represent a powerful technique for the analysis of agricultural data. In this work, I think that we have gone one step further, and I hope that I have been able to show you that PSPLANs can also be useful for the analysis of high-throughput phenotyping data that we can obtain from the model interesting insights. This model has a drawback. Although we have solved part of the computational aspect, there are still some problems going on. One of the problems is that the model does not scale well particularly if we want to be very generous with the B-spline basis. And this is especially important if we want to be very generous with the number of B-spline bases that we use to model this spatio-temporal surface. So one way forward would be to leverage sparse mixed model formulations, and I would recommend you the reading of this paper by Martin Burr, and also the package that it's described there, because it's really, really nice, this work. So this will be perhaps the first thing to try. Of course, this work has several limitations, as well as many areas that I believe deserve further work, and I won't go through them, but here you have some of them. So with this, I finish. So thank you very much for your attention, and if you have any questions, hope I will be able to